Frederick Griffith was a British bacteriologist. In 1928, Griffith discovered bacterial transformation, where a bacterium distinctly changes its form and function. To prove transformation, he did an experiment with pneumococci. In this experiment, Griffith used two forms of this bacteria, rough and smooth. He conducted his experiment using mice as his subjects. Mice injected with R cells remained healthy. When Griffith examined blood samples from these mice, he found no live bacteria. Mice injected with S cells developed pneumonia and died. When Griffith examined blood samples from these mice, he found live S cells. Mice injected with S cells that were killed by exposure to high temperature remained healthy. When Griffith examined blood samples from these mice, he found no live bacteria. Mice injected with a mixture of live R cells and heat-killed S cells developed pneumonia and died. When Griffith examined blood samples from these mice, he found both live R cells and live S cells. Heat killed the S cells but did not destroy their hereditary material, and that material was transferred from the dead S cells to the living R cells. This is how the concept of transformation came about. First, the donor DNA fragments bind to the proteins on the cell surface. Then, donor DNA fragments are transported into the host cell and become single-stranded. After that, there are two possible outcomes. The first one is a successful transformation, where the single-stranded donor DNA fragment is incorporated into the host chromosome. The second possible outcome is an unsuccessful transformation, where the donor DNA fragments are broken down and are not being used by the host cell. Transduction is the process by which DNA is transferred from one bacterium to another by a virus. There are two types of transduction. In general transduction, any of the genes of the host cell may be involved in the process. In special transduction, however, only a few specific genes are transduced. In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called bacteriophage or phage. The phage attaches to the bacterial cell and injects nucleic acid into the host cell. A phage enzyme is reproduced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and the phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of the phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material. Transduction and specialized transduction are especially important because they explain how antibiotic drugs become ineffective due to the transfer of resistant genes between bacteria. Transduction was discovered by Norton Zinder and Joshua Lederberg at the University of Madison, Wisconsin in 1951. However, the mechanism of general transduction, as well as specialized transduction, was discovered a few years later by Esther Lederberg, Larry Morse, E. Lively, Alan Campbell, as well as Joshua Lederberg.